Okay, we're good. Good afternoon to some of you. Welcome back. To some newcomers who are just joining us, my name is Thea Guidry, and I run the GeneLab core lab housed in the School of Veterinary Medicine. GeneLab provides a variety of molecular services, and we try to invite speakers in to share with our community different techniques and technologies available. GeneLab is part of the LBRIN Molecular and Cell Biology Core. In keeping with our mission of inviting in speakers, we have Mark Lawson here with us today. He's the Field Application Specialist for Bio-Rad Laboratories. And he's going to go over session 3 of our GeneLab Molecular Biology Training Course series. This session 3 is information on the BioPlex Multiplex Immunoassays. And Mark, let's go ahead and get rolling.
Great, thanks. So I'm going to be, again, up and down a bit. It's fun for the camera person. So we're going to be talking today about the BioPlex Multiplex Immunoassay. It's both a platform and a chemistry. And some of you might be familiar with it. There are a number of units throughout Louisiana. I know at the LSU Health Science Centers, both in New Orleans and Shreveport, they're pretty heavily used. So before I get really launched into my presentation, from the satellite sites, if you guys could let me know with just a hand raise, how many of you are doing ELISA or any kind of protein assay? ELISAs? No ELISA people. All right, how about if you raise your hand if you aren't currently doing ELISAs? Aha! That proves it. They're both doing and not doing ELISAs. All right, so how many of you are doing flow cytometry? How many of you are not doing flow cytometry? Okay, all right. Now we're getting it. So this technology combines elements from both flow cytometric analysis and ELISA-type protein assays. It has the capability to change the way that we ask questions about especially the immune system and the response of the immune response to various challenges. It also has applications for phosphoprotein cell signaling. It also has a number of other applications. It's a very open type of platform for the development of new assays. And a number of companies have responded to that by the development of specific tests. So how does it work specifically? The hardware here is manufactured by a company in Austin, not too far from where I live. The company is called Luminex. They've been around for, I think, 2 decades, about 20 years. But they've only in the last 15 launched this platform, the hardware. And it is a multiplex platform that incorporates the flow cytometric and ELISA type assays. They make the hardware. Bio-Rad takes their hardware, loads their own software, and sells some supporting equipment that makes it a little bit more useful for the end user. So what exactly does it do? It can supplement or replace. And we honestly see it more of a supplementation rather than complete replacement for most cases. Supplement or replace our Western blots, ELISAs, protein arrays, macro arrays, we're talking about protein primarily, and PCR-based techniques. There are a number of, for example, genotyping applications for the genotyping of lots of different SNPs or markers simultaneously in the same sample. It increases the scope of study, and that's the most important thing. Again, changing from the way we're currently doing things, which is primarily driven by a single hypothesis. We test a single hypothesis, and then we determine whether or not that hypothesis is supported or not. This changes to something where we're screening lots of possible relationships at the same time in order to find the ones that are interesting for further study and analysis. Right? Decreases cost versus a number of other techniques that are more labor-intensive and also cost-intensive. So it is a shortcut on cost. It's also a shortcut on time. We increase the throughput of samples that we can process in a very short period of time. So you're looking at about 10,000 data points per day without breaking a sweat, with as little as 12 microliters of serum per sample. We have an increased sensitivity and linear range when compared to a number of other more labor-intensive methodologies. So for a cytokine assay, we might get anywhere from less than a single picogram per mil to 32,000 picograms per mil. How does it work? Well, we have to back up first and talk about how a traditional ELISA works. We have, let's say, a polystyrene well. I'm showing one well out of a 96 well plate. I'm switching colors. We start with the polystyrene well, and to that we couple capture antibodies. These are antibodies that are actually bound to the polystyrene. Okay? We add our sample to that well, and it's going to capture the analyte from the sample. So this could be, for example, IL-2, right? Or IL-6 or IL-8, or whatever type of analyte we're looking to detect. We then add a detection antibody to create an immunoassay sandwich, right? With the bread being this antibody, and our meat and fillings being the analyte. And I've drawn this so that they're sort of monovalent antibodies rather than divalent. But the whole point is that we have a sandwich of antibodies detecting the analyte. On the capture antibody, we also have a reporter molecule, usually HRP. I think that's one of the more common ones these days, which is, I think, a horseradish peroxidase. And in the presence of a particular substrate, we get a color change. And that color change reflects, changing again, reflects the amount of analyte that was detected. So we simply read this from above with a light signal, incident light comes in, and then we read the return light, and we figure out how dark or how opaque the solution is, right? And that's how we quantitate that, okay? That's traditional ELISA. Let's switch over to the xMAP technology that the BioPlex is based on. Instead of a well made of polystyrene, we still have a 96 well plate, but the polystyrene to which we attach the antibodies is a bead. It's a 6.5 micron bead. So put that in relation, that's about the size of a red blood cell. So we have a very tiny little polystyrene bead, and we use the same chemistry to bind it to the capture antibody. So we now have a bead coated all the way around. You can draw the other ones. It's like a little sun. Coated all the way around with our capture antibody. And instead of adding the sample to the well that's already pre-coated, we can add this directly to a sample. So we take our sample, we add the beads, and instead of interacting in a flat surface, we're now going to be interacting in 3 dimensions, right? We've got our 3 dimensions. And the interactions are occurring as they're shaking. So we move the sample and the beads through each other, essentially, to get maximum interaction. We're adding detection antibodies as well. So we've created our immunoassay sandwich. We've got our capture antibody and our detection antibody. And the only other difference here is that this is a biotin. So we're using a biotinylated detection antibody. And this is a strep avidin, or I guess it would just be, sorry, PE. A strep avidin phycoerythrin. Phycoerythrin is a very common flow cytometric dye. So it's just a fluorescent molecule. So at this point, what we've really created is a FLISA, right, which is a fluorescent linked immunosorbent assay. And it's for one analyte. That's okay, that's pretty cool. So we've made it fluorescent instead of enzymatic. We run this through a bead sorter or a bead reader, right? A very specialized flow cytometer. And that laser is going to come out and it's going to read for the presence of RPE or phycoerythrin as each bead is passed one by one through a flow cell, right? So the beads are forced into a single channel and as they pass through, they're being interrogated by a particular laser. But that only got us as far as the ELISA. The strength of this technology is that the beads themselves are different colors. So we can have 100 different bead regions. To each of them, we attach a different kind of capture antibody. This one, let's say, is IL-2. Right? This one is GM-CSF. Whoops. And this one is, well, let's do IGF. Keep forgetting. Whoops. And this one is IGF. Okay? So we've got 3 analytes in the same well at the same time. We're going to analyze a single sample with all 3 beads. So now, depending on how many of each of these analytes is bound, let's say we've got some analyte here with our detection antibody, same thing here, right? As each of these is run through the detector, not only are we reading how much analyte was detected by that bead, we're also analyzing the identity of the bead itself. So as it runs through, we say, that's the GM-CSF assay. Let's read its results. That's the IL-2 assay. As it runs through the flow, I can say, those are my IL-2 results. So even though they're all mixed up together, 3 beads all mixed up in the same well, I can separate them as they pass one by one through the detector. So here are the 100 distinctive beads. We have 10 concentrations of red. This is just to make you happy, I want you to know. And 10 concentrations of infrared. So with all the combinations between those 10 concentrations of red and 10 concentrations of red, we end up with 100 distinct bead regions. And in fact, we number them from 1 to 100. So as they pass through the flow cell, we assign the results to the correct window. And this, for example, might be that's our IL-2 result, and the results coming out of 83, that's going to be our tumor necrosis factor alpha. All right? So with 12 microliters of serum, I can analyze up to 100 different cytokines and chemokines simultaneously without having to run, well, let's flip to the next slide, without having to run 27 separate ELISA plates. I can run a single xMAP assay and do the same amount of work as I would have done running 27 plates. Now, I don't know anyone that would willingly sit down and do 27 ELISAs. I think that's probably going to be rate limiting. It's going to be expensive. You're looking at $20,000 for that kind of data. And you're looking at 60 hours or more, which of course is a lifetime in grad student terms. And the total sample used would be probably a full vial or several vials worth. You don't always have that as an option, right? So very limiting, and that's why we might only look at this one and this one and this one. We might only look at 1, 2, or 3 targets because everyone kind of knows that this is TNF-alpha mediated, and I just want to verify that. With the xMAP, I can find out which ones are actually modulated in my disease model, but I can also see which ones aren't modulated. And that sometimes is just as important as seeing the non-stimulation of a particular pathway. So we've gone from hypothesis-driven to screening. And maybe the screening is upstream. So you do your xMAP plate, screen all 100 analytes, and then when you find the ones that are interesting, you continue to screen them by some other technology. Although a lot of people don't. A lot of people stick with it all the way. It's also more sensitive. We don't really push that angle. It also has a broader detection range, but they're very small incremental improvements over existing technologies. For example, it's more quantitative and reproducible than a Western blot for phosphoproteins. But it's not that big a difference. So the key difference is the ability to multiplex to very high levels. But it also improves the quality of the data somewhat. Now, we've got the cytokine assay that we've been referring to so far. But we can think of this more generically. We have an antibody capture step. we have the analyte, and then we have a detection technology, which again is an antibody. But it doesn't have to be that format. You can do the same thing with the same bead. You can have the capture technology be a nucleic acid, be a transcription factor, be a receptor. Anything you want to attach to a piece of polystyrene bead can then be the capture technology for an analyte. In this case, here's a simple microarray style nucleic acid quantification. We've got a capture oligo bound to the bead, and we've got a biotinylated PCR product, which is simply created by using a biotinylated primer. And we can use our RPE label for 100 different capture beads and determine the presence of a particular nucleic acid in a sample. So you could use this to interrogate bacterial populations. You could use it to detect a variety of different mutations, antibody specificity, receptor interactions. Really, with the generic idea of capture and detection bound to the bead, you can do a lot of other things. You can do, for example, enzyme specificity, protease specificity. You can do receptor-ligand interactions, right? I've got a histone receptor and I want to know what molecules it's most specific for. I can actually do that with fluorescently labeled ligands. Any questions about the technology?
Yes.
Okay. Yes, good.
Okay. If I understand it correctly, the ELISA can actually distinguish the identity of those different color beads. And I thought the ELISA, sorry for my ignorance on that. I thought I'm sorry. You're talking about the flow cytometry. I saw the flow cytometry actually distinguish the size. Okay, so let me rephrase the question. For the different beads with different color, when they go through the flow cell, they will be identified. How would they be identified? Just because they have different color?
Yes.
Or because different size?
Just different colors. You may be thinking about another technology called CBA, which is it's a Beckett Dickinson specific technology. And they use different bead sizes. All right, and their level of multiplexing is usually constrained to 10 or less. I think they've got a few that are above 10 or 15.
And so the flow cytometry distinguish different beads. What's the mechanism of distinguish the color?
They're actually fluorescently doped. So at the factory where the beads are made, they basically synthesize them in an organic solution, and the polystyrene swells up. And then they add the dyes to the organic solution and change it out to an aqueous solution, and the beads shrink back down. And they capture the dye that was present in the solution at the time. And so all you have to do basically is cook them in phenol, add the dye, swap it out to water, and then those beads are labeled with a very specific fluorescent signature.
Okay, so for example, for the IGF, if it's labeled with particular fluorophore, the emission color has to be the same as the color of the beads?
Oh, I see. The label on all of them is always RPE.
Okay, I see.
So the only thing that differentiates them is the dye that's actually inside the bead.
Okay.
So if you want to imagine, we're reading 2 channels for identification and 1 additional channel for the signal, for the reporter, right? So we actually call it the reporter channel, the RP1. As the beads are coming through, we gate the results. If you've done flow cytometry, we create gates, right? In each of these gates, we read the results for RP for the beads that match that window, right? So based on the windowed analysis, we can segregate our results into the kind of assay that it is, right?
Yeah, makes sense. Thanks.
We do also analyze the size of the beads, but that's a QC procedure. And we do that through essentially a side scatter analysis, which we've called the doublet discriminator. And the total system, and I'm never clear on this in my presentations, but the system itself is hardware plus the bead systems, which are labeled with assays. So what Bio-Rad sells is the hardware, the software, the analysis, the controls, QC controls, and also a number of assays that are already set up for, I think our total right now is 50 human cytokines and chemokines. Other companies also make assays for the same platform. So when you buy any instrument from Luminex, one of their distributors, they're interrelatable. So you can run a millipore assay on a Bio-Rad instrument, and you can run a Bio-Rad assay on a millipore instrument. All right. Did I answer?
Yes, thank you.
Okay.
Mm-hmm.
Anyone here have questions? Okay. I wanted to put a couple of things together on best practices, especially if some of you might have access to a BioPlex and want to give it a try. The format for my presentation today, I want to tell you a little bit about how to contact us, how to start up and shut down the instrument, how to maintain, calibrate, and validate the system, how to prepare your samples, read your samples, analyze the data, and report the results. I think those are the key things to talk about. So contacting us, we have a tech support. This is also true for our real-time, that's accessible from 1-800-4-BioRad. If you hit extension 2, that takes you immediately to our tech support team. So our real-time amplification group, I believe, has 11, and they're all PhD, they're essentially all postdocs. And we have, I believe, 4 or maybe 5 right now on the BioPlex team. If you hit extension 2, that will take you right to a human being. We wanted to avoid the whole press 7 if you speak English, press 6 if you are calling from a rotary phone. That's a fun one. We wanted to keep it as simple as possible, and they really are quite good, and they're available for Louisiana time, 7 a.m. to 7 p.m. So if you're in the lab outside those times, you should not be. We also have field application scientists like myself. We have account managers and instrument specialists as well. So we have quite a team. And in Louisiana, you also have Randy Thiaville, who everyone seems to know because he gets around a lot. He's been here for, I think, 2 decades. He's been here a long time. He keeps moving up in the company, so he's a regional manager now. So the system has some automated routines. Here, I'm throwing the camera off again.
Yeah, that's
Sorry. Has some automated routines. Now, it does take 30 minutes to warm up, so it's something you want to plan ahead. The prep usually takes about 2 hours. The run takes about an hour, and then the analysis takes about 5 seconds. So we start with a startup routine, which runs some organics through the system to debubble it, rinses the needles off, and then we have a shutdown procedure at the end of the day that will actually run bleach through the system to keep it clean, sterile. I'm not quite sure what the proper term would be. It's very important that you run these things to keep the system from getting damaged. Calibration, because it's a laser-based system, those lasers over time become less bright. So it's very important that we correct for the drift in the lasers, the optical decrease in brightness. For that reason, we have beads. These are the same kind of 6.5 micron polystyrene beads that we run through the system with very fixed fluorescent properties. So what that has the effect of doing is making small micro adjustments to the sensitivity of the detectors, so that as the lasers age, we boost the sensitivity to compensate, so that the same results you're getting today will match the results that you're getting 6 months from now and 6 years from now. So running that before every run also acts as sort of a dry run of the instrument and verifies that everything's operating properly. So we recommend running a calibration bead run right before each plate. To calibrate the system, there's a software routine, and you input those fixed values in a dialog box, which then tests it against the known values. Now, that's the daily operation. We also have a more complicated validation procedure, not complicated, that tests a lot more elements of the system. So it tests, for example, any bead carryover. It tests the accuracy of the detectors. It tests the ability of the system to span a particular dynamic range. So anytime you want to do troubleshooting, or if you want to make this part of a routine operation, you can do this on a routine basis. There is a sample needle that represents the weakest point in the system. And it's the needle that dips down into the samples and aspirates the sample up. If you've got a very dirty sample, especially if you're working with certain serum or plasma samples that contain a lot of junk in them, it may be occasionally necessary to replace or clean the needle. And so the way we know that is when we run our calibration procedure, it will tell us in this field how many beads per second are passing through the flow cytometer. If that number drops below 150, it's probably time to replace it. And this is a sort of a fun procedure because it makes a lot of crunching noises. You rip out, I'm using the correct term here, you rip out the light housing just above the needle, unscrew the needle, and then pop it up from underneath. Then you can take just a squirt bottle of water and force whatever it is that's plugging up the needle. The camera keeps adjusting on me. Plugging up the needle and squirt water back through it, and that usually unclogs it. And if you'll do that before you run your precious samples through, you'll avoid losing any data. And then once you've replaced the needle, it's sometimes necessary to adjust the needle height. And to do that, you just loosen the screw, and then there's a calibration plate that we're going to use that will tell you when the needle's adjusted correctly. So that's really it for hardware adjustment. Everything else relates to the sample prep. And I've got a couple of tips in case you guys ever decide to use the BioPlex systems. These are best practices. The very first thing, and I think this is true for ELISAs as well, is to map out your plate on paper. Make sure you know where the samples are going to go before you start thawing out your samples and pipetting them onto a plate, because it's very easy to get confused once the samples start getting transferred. And part of that is deciding in advance if you're going to have space on your plate for all the samples you intend to run. Right? And that may even involve mapping out an entire set of samples in advance so that you've thought all this through. Want to make sure you've reviewed the protocol before you start. So that means that you know what kind of volumes you're going to need, what types of pipetters, what kind of pipettes. Clean your work surface. This is something that's important to avoid contamination. Always calculate excess anytime you're using a multi-channel pipetter. That's common sense. Include blanks in at least triplicate. So this assay has a certain amount of background. As you might imagine, any antibody-based test is going to have a certain amount of background signal. Knowing what that signal is very accurately will help us to subtract that value out when we do our analysis. So I always recommend at least triplicate blanks. Bring all the reagents to room temperature before you start putting it in the machine. And that is especially because of the problem of bubbles. Bubbles, as they pass through the flow cell, have a tendency to look like particulate matter, or something actually gets registered as the lasers shine through the bubbles. They create little patterns. So we want to use warm reagents or room temperature reagents during the run. And of course, if you make any changes to protocol, you want to make sure you document that. That's good lab technique. The procedure for preparing the samples. The very first thing we're going to do, we're going to take a generic filter plate. Or if you're using the latest technology, is a magnetic prep. And the beads themselves for all our cytokine assays are both compatible with a magnetic prep and with a vacuum prep. And I'll talk about that a little bit more in a minute. Allow yourself about 2 hours for the prep. And so that means if you're planning on eating at noon, start before 10 a.m. So we're going to add the beads to a generic plate. It's either going to be a flat bottom plate or it's going to be a millipore filter plate. And that filter plate has a membrane on it that allows, it's got a 1 micron pore size. So as you already know, the beads themselves are 6.5 microns. So they're not going to be able to pass through the pores, but the 1 micron allows the liquid to pass through. So the beads stay in the well at each vacuum step, but the liquids get removed. So you want to make sure that if you're going to use vacuum, that you've calibrated it to not over vacuum the very thin membrane that is the 1 micron selective filter. Okay? Vigorously vortex bead solutions because the beads have a tendency to settle. If you let them sit for more than 30 seconds, chances are the beads are starting to settle out of the solution. Transfer the beads to the vacuum filter plate. So at this point, you have a filter plate, again, a generic filter plate or flat bottom plate, and we've transferred the beads, which contain the capture antibodies, onto that. We're going to wash those twice with an assay buffer, which contains protein. That's specifically to act as a blocking agent. Then you're going to prepare your standards and samples. And this is where most of the work happens. The best thing you can do is to have two plates. Plate 1, and yes, I'm loving having something to write on here. Plate 1 is where you create your dilutions of sample plus a diluent. All right? Plasma and serum usually work best if you dilute them 1:4. Then we're going to create plate 2, and that will have the beads in it. And we're going to transfer using a multi-channel pipetter the samples on top of the beads. And the reason why is the incubation times are very short. And someone with a single channel pipetter to transfer 96 samples or 96 wells worth of sample will take a long time, right? It usually takes 30, 40 minutes. It's a 30-minute incubation. So you would end up with a 30-minute time differential across the plate for a 30-minute incubation. And that creates a lot of variability. So the very best thing you can do is have your samples already arrayed and use a multi-channel to transfer them to the actual plate. And that means you can transfer all the samples in less than 5 minutes, so there's not a big differential in time. The standards are, there's a protocol for creating sort of normal standards. We have them both at the high range and the low range, depending on whether you're looking for sensitivity. Kink in my shoulder. But you can change that to meet your own needs. We have it currently set where it's the broadest possible range. You may want to pre-filter your samples or at least spin them down. If you get enough debris in the samples, it can actually interfere with the instrument. It can get clogged up in the needle. We'd like to avoid that. That's also true for hemolyzed or hyperlipidemic samples. Serum is best in a tiger top tube, and plasma is usually best in a sodium citrate tube. If you're going to use unusual samples, and we've got good data on urine, CSF, bronchoalveolar lavage. We did tears once. That was an interesting one. And we did some fecal samples that didn't work all that well. So we know most fluids work pretty well. Some things don't contain a lot of cytokines. We've also done microdialysis of brain fluid. Always a lot of fun collecting the samples. If you've got a sample that doesn't contain a lot of protein, it's sometimes necessary to add a certain amount of blocking or carrier protein to the sample to prevent that very minor amount of protein from sticking to the sides of the wells. So it may be necessary to add up to 0.5% BSA. You always want to protect these reactions with aluminum foil, protect them from light, because the beads themselves are fluorescently labeled, as are the detection reagents. We don't recommend belly dancer shakers. Those are the ones that sort of wiggle around. We need something that vigorously shakes the beads, again, to keep them from settling out of the matrix. And the fact that they're interacting in 3 dimensions is what gives us the boost in sensitivity versus a standard ELISA assay. And make sure you use a lab timer. We find that people trying to mentally think of what 30 minutes is have a very poor perception, depending on whether they get an email that they really have to read or they're just sitting there staring at the wall, where 30 minutes can seem like forever. You want to use a multi-channel pipetter whenever you can, again, because of the time differential. And you want to be careful to seal the plates so that when we go to vortex them or put them on a very strong shaking platform, that we don't end up with liquid splattered all over. We have had some cases where people couldn't understand why the volume kept disappearing. And it took us a while to figure out that they hadn't actually sealed their plates. They were putting them in the vortexer, vortexing them very vigorously, and there was a coating of sample on the inside of that chamber. And then the final step here, after we've washed the detection antibodies away and we've washed the sample away from what's not bound to the primary antibody, we're going to add strepavidin phycoerythrin. And that's going to be part, depending on how you order the kits, of one kit where we now have an all-in-one format. And then you resuspend the beads right before you read them, put them in the system, and then the system reads all 96 wells in an automated fashion. And it can skip wells, and it can read them in whatever order you like. A couple of very important sample prep tips. Number one, and I think we've emphasized this already, the uniformity of the prep is very important. If you have the big time differentials, that creates a lot of variability. It's also true that if you keep switching pipetters at each step, you'll introduce a lot of variability as well. Filter plates are very delicate. If you over vacuum them, they can actually rupture. That's not true of the flat bottom plates, but it is true of the millipore filter plates. The beads settle over time, so again, if they sit more than 30 seconds, we want to read vortex. And we want to lay out the samples in the 96 well format before transferring. So that's where I said we had two plates. We have a prep plate and we have the assay plate. We're going to transfer the samples to the assay plate with a multi-channel pipetter. And then lastly, the membrane, for this slide, the membrane on the prep plate itself is permeable. So we've had a few cases where people would put their prep plate down on a paper towel and would actually wick the liquid down through that membrane, and they would come back to a completely dry well. Amazing how that works. The strep PE is especially light sensitive. If you're working in a nice, brightly lit, sunny lab, good luck for you, or good for you. But that also means that you need to be especially careful about bleaching out your strep-avidin PE. The polystyrene binds proteins non-specifically, and that's why we need at least 0.5% protein if you have to add BSA to prevent that from being a problem. Always prepare excess. A lot of cases where you'll end up under pipetting because you're using a multi-channel in a reservoir, you tend to lose quite a bit in that transfer. Always run in replicate if you're going to be concerned about quantification. And then multi-channel pipetters can create a lot of inaccuracy. If you haven't already experienced this, you may have noticed that when you pull up liquid, there's always one channel that doesn't equal the others. So reading your samples, this will usually be specified by the kit that comes in. Right now we're recommending about 50 beads per region. So that means for each of these different bead types that we have in the well, we're going to read 50 beads. That's 50 events that we're measuring. And those 50 events will give us a median fluorescent intensity. So we refer to that as the MFI, median fluorescent intensity. If necessary, if the fluidics for some reason become clogged, it is possible to designate certain wells to be reread. And that's not at all uncommon, that you may want to reread them at a different level of sensitivity, or you may want to reread them to confirm the results. In that case, you simply put a check in the box that says rerun or reread. In analyzing the data, we have a number of different things that we can turn on and off that might be relevant. Part of this, of course, is quality control. So we can turn on the raw bead statistics, which will tell us, how many beads we had collected for each assay. So you want to know that for your IL-2 assay, you've collected an adequate number of beads to be a statistical sampling. And we've said it again, not to move on to the next well until we have at least 50 of each bead type. The other thing we're collecting as the beads pass through the flow cytometer is we're reading what we're calling the doublet discriminator. And this is a side scatter analysis of the bead events. If the beads are the correct 6.5 micron beads, they will fall between those two red gates, right, which are shown as dotted lines. If for any reason two beads happen to be stuck together, popping up again, if the two beads for any reason happen to be stuck together, you'll get doublet peaks. And those are shifted to the right here on the doublet discriminator. And then a triplet peak might be all the way at the edge of what we can actually detect. Now we are filtering that data out, or we would say gating that data out. So the gates for the data are between here and here, and these are the single bead events. All right, so that's the only part that goes on to the next step. And that's an important part of quality control. If for any reason your beads get jammed together, and one common thing is fluidic problems. The beads are stuck together. They're still big enough to pass through the flow cell, right? And if that happens, you'll get twice the fluorescence that you need. The other thing, once we've gated the data, what is gated comes then over to the region or the bead map, which has 100 regions on it. This is the classification 2, that's our infrared dye that's doped into the beads, and the classification 1, and that's our red dye that is doped into the beads. So this is identifying which region those beads are assigned to. So as they come through, we're reading them, they're the correct size to be a bead, and they correspond, let's say this is region 17, and that's our GM-CSF. Right? And then what fits in here will then take the reporter signal, the RP signal, and we'll measure that and compare it to a standard curve and determine the amount of GM-CSF in that sample. Right? But if we get events that are outside the specific regions that represent the well-defined beads, then it might be debris, or it might be bubbles that happen to be about 6.5 microns or have the same apparent size as a 6.5 micron bead. So it's very important that that stuff be removed before we do the analysis. If the samples are photo bleached, and this is very unusual, I've only seen it once in 4 years, all of them will shift down and to the left. And that shows that you've photo bleached your beads. So all of them shift down and to the left. And that will get flagged as a big problem. There's really no way to recover from that. In analyzing your data, here's what a tabular format data would look like. It tells us switching colors, the type of sample, so that's standard number 1, that's the highest abundance standard. Then we get the median, terrible underlining, the median fluorescent intensity, and then in parentheses we get the number of beads that represents. So the median of 100 beads, 146 beads is 13,415. That was the median fluorescent intensity for the beads that were gated and properly regioned for IL-1 beta, okay, for mouse. All right, we're also getting the percent aggregated beads. Those are the number of beads that were stuck together. And if we do our prep very poorly, we'll sometimes find that number goes above 10%. In this case, we have a very high abundance. Cool. It has a very high abundance, so some of the beads did actually end up getting stuck together. A couple of other values that are important. We have the Fi, which is the fluorescent intensity. We have the Fi minus the background signal, and that's what we're mostly using with our samples. We get standard deviation, percent CV, observed concentration, and expected concentration. So that's what we typed in. Oh no. There. The expected concentration, that's what we typed in. And this is where it fits on the standard curve model that we just created with these standards. So then we calculate, and this is very important, the observed divided by the expected times 100. In other words, I expected the value to be 10,000. That's what I typed in. And the actual value, when I put it back on the standard curve, is only 9,500. So that is 95% of the observed divided by the expected times 100. So that's a way of testing whether or not the standards are good fits for the standard curve model that we created. Right? And we set early on a range for what is acceptable. And by default, it's 70% to 130% observed divided by expected times 100, which we often call the spike recovery or the recovery. And so that tells us how close we are to the expected values of the standards. And that is a very important quality control for whether or not our standard curve is accurate and whether or not certain standards should be on that curve or shouldn't be. All right, concentration and range is the values without extrapolation. We'll talk about that more in a few minutes. If I were going to hand these results to a clinician, I would only be using the concentration in range. All right, the software that we use has a built-in 5-parameter logistic regression. And again, I don't pretend to know exactly what that means, except there are 5 parameters to the curve fit. And that includes things like the baseline, the upper end, the slope, the drop, and then also the asymmetry. So the 5-parameter logistic regression does a very good job of matching this data. Now we're graphing it in a fairly standard way for us, which is the concentration is on a log scale, and the fluorescent intensity is on a linear scale. Most people, when they first start out, prefer to see log-log comparisons. And we give the equation for this particular data. Now, in order to do a 5PL comparison, we need 6 data points. So a lot of kits that you'll buy from people other than Bio-Rad, they recommend a 6-point standard curve. The problem with that, of course, is it prevents you from dropping any data off without dropping to a less appropriate curve fit model like a 4PL. So going back to recovery, it's a very good way of estimating whether or not our model works over the range that we're testing at the time. And we can adjust that to be acceptable or not acceptable, and that creates a certain amount of automation. So we want to automate accepting or rejecting our standards as being part of a good model or a bad model. And in this case, you can see I've got some values for recovery, or observed divided by expected times 100, that are way outside that 70 to 130 range. And that's because they don't fit on the curve very well. By dropping those off, I'm going to end up with a better analysis. So this is the one thing that we end up having to do somewhat by hand, except in the very latest version of the software, we do actually have an algorithm that does this on an automatic basis. Now, some of you that might have done ELISA in the past will be familiar with what's known as the prozone effect, or what I've come to call the hook effect. And that is, I've got increasing amounts of concentration as I go across, and I've got increasing amounts of fluorescence. And you can see as I increase the concentration, I get an increase of fluorescence up to a point, and then it suddenly hooks back down. And that's because I've saturated all the antibodies. And as I continue adding more analyte, it actually becomes inhibitory, so that I get less binding of detection antibody. It actually soaks the detection antibody up and pulls it out of the reaction. So we go beyond the saturation level for the antibodies, and we start to get a decreasing return of fluorescence. And when we include the points that are on the hook, the 5PL can't model that very well. So we end up with a bad curve fit. The best thing to do is to cut these things off, and I'm showing it here in red, so that you drop this point off and you drop this point off and you get a much better curve fit. And that improves the quality of the data for the rest of the range. So we won't get data above 10,000 picograms per mil, but we will get better curve fits down here at 2 and 3. And it is possible to exclude the data. Here, where we have an outlier column, you can do partial or total outliers, depending on what you think is appropriate. Of course, there's always what I call the postdoc's dilemma, which is how much can I cherry pick my data and still have it be valid? Right? At some point, what do they say? The R squared between any 2 points is 1. So if you drop enough points off your curve, eventually your R squared will be 1. And so that's that same hook data. Remember where it was going to hook around. And if I go through and I drop those points off as outliers, I get a much improved curve fit all the way down the line. So even though I've sacrificed the upper end of this particular assay, I've greatly improved the lower end, which is maybe where the more exciting things are going on. You can also do partial replicate outliers, and that's, of course, appropriate if you have a particular outlier. One of the things I always go through and screen for is very high CVs. If you see a CV above 20%, that's usually a flag that maybe you missed that one with the pipetter, or you jogged your elbow a little bit, or wasn't prepared correctly. And it's a good idea to go ahead and drop the outlier. If we're going to do that for the blank, I mean, here I've got an example where the values were 185, 45, 7, and 7. With a quadruplicate, it's pretty obvious to see which one of those is the outlier. I mean, maybe not statistically, but I feel pretty comfortable saying, you know what, drop the 185, okay? And when I do that, it improves the sensitivity of the total assay. So it's very important that you have enough points where you can do that justifiably. All right, now it will occasionally give a value of out of range. So out of range means that the fluorescence reading, for example, is negative, or that it's so far below the lowest standard that we're not able to extrapolate. So out of range less than means below detectable limits. And what you do with that in your statistical test is entirely up to you. There's been a lot of discussion of that lately.
Well, could you get, if you end up something, if you end up dropping off data points on the upper end of your range so that you have a better fit, could you end up with one sample that's actually more concentrated than your highest standard at that point?
Yes.
How does the software, is it still indicated with an outer range? What would show up in the software?
I'm glad you asked, because it would either be out of range if it's more than 2 standard deviations beyond the highest, or you get a star value if it's willing to extrapolate. Now, star values are basically extrapolated points. So we've taken, let's say in this case, and it's basing it always on the fluorescence, not on the concentration. And that's usually because we're multiplying the samples times 4, because they've been diluted 1 to 4. So here I am, I'm at 35 with a standard deviation of 1.4. However that worked out, that was within that range, just barely. And it came up with a star value, but you'll notice that does not transfer to the concentration in range. So that's a way, if you want to know what the value would have been, if we extrapolated it out just a little bit, you can get the value. But when you go to hand it off to a clinician, you hand them the concentration in range, and you force them to not process that information. Not to insult clinicians.
Well,
yeah, go ahead. Exporting the data is fairly simple. It's just a matter of clicking on the Excel icon. And you can export that in any format you like. I won't go into detail. We do use a stat-LEA 5-parameter logistic regression. It's a very complicated equation. It accommodates, again, the low end, the high end. All you really need to know is that it accommodates an asymmetry that's very common in these types of assays. So it does a great job of curve fitting. Some would say too good a job of curve fitting. If you have any 6 data points, you can usually come up with a pretty neat function. We are very careful not to do this. Some of our competitors are. So we try to educate people on the idea of creating a 0 point on your standard, where you use essentially a blank and call it a 0 concentration standard. And what it ends up doing, of course, is extrapolating from the last measured down to a 0 value. It's called forcing the function. And it creates lots of model information between your lowest standard and an arbitrary 0. The problem is it's not based on an actual measurement. It's a very unreliable way of modeling data. So we really don't recommend that, using 0 points on your standard curve. I'm going to skip a few things here. Exporting to Excel, you probably want to export in a variety of formats. If you're going to do sort of a meta-analysis in Excel, you're going to analyze your data a little bit further, having it in the format of IL-1 alpha, IL-1 beta, IL-2, IL-3, IL-4, IL-5, all on one sheet, with all the extraneous information kind of trimmed away, like CVs and standard deviations. Just those values in a single sheet allows you to do a lot more batch-type manipulations in Excel. But we also offer the ability to export the full information about each assay as its own tab. That just requires more effort if you're going to be doing any further analysis. You can also copy the graph with a lovely soothing blue color. It's good. That's a psychology thing. It keeps you from being unhappy. And then let's talk real quickly about how to order one of these assays. So the first thing you need to do is locate where the instrument is at in your account or your institution. We'd be happy to help you do that. I don't think anyone on this call doesn't have access to one. Maybe Louisiana Tech. We'd have to check. But they are in a variety of different places here in Louisiana. We've got probably 8 in New Orleans, several on this campus. And there may be some older ones I'm not aware of. I've only been with the company for 4 years. We've been selling this for 10. So you would go to our website. We have a number of pre-made assays. And those are things like a panel of 27, a panel of 23. But you can also order up custom mixes or individual items. So you go to our website, and we have an xPlex assay website. You put a tick in the box for the ones that you want to include in your panel. Now a 27 plex costs about $2,000 for 96 results, one plate, which is a little intimidating. A lot of people that are used to paying $300 for an ELISA suddenly go into sticker shock when they're presented with a $2,000 price tag. But remember, that's 27 ELISA kits. So on a price-to-price basis versus a pre-made ELISA kit, we're usually about half to a third of the price of a pre-made ELISA. Now, we can't always compete with the do-it-yourself ELISA kits where they send you antibody and you couple your own plates. You have the choice of ordering these things pre-mixed, and the only people who do that are people who don't like the effort of blending their antibodies together, and we charge a premium price for that. Or we'll send you the individual vials for, let's say, a 12-plex, and you blend those 12 components together. Right, and that creates your 12-plex multiplex immunoassay. The kits now come in an all-in-one format for cytokines, and everything you need is in one box. So it contains the wash buffer, assay buffer, the SAPE, the standard material is supplied with the kit, different diluents, and different detection antibody buffer. And then this will be your capture bead, and this will be your detection antibody. So that all comes in one kit. So here are some of the targets that we have. Again, we are only one of about 7 manufacturers of kits. We focus specifically on cytokine and phosphoprotein kits. We also have some special disease panels. But you can go to luminexcorp.com, and they've got a comprehensive list of all xMAP compatible assays. So here are some of the most popular targets that we decided to offer in our first panel, and that's a total of 27 different analytes. And then we also have a human 23-plex for a total of 50 unique targets that can actually be multiplexed together. The only ones that don't give good multiplexing data when they're blended is IL-12P40 and IL-12P70, because those tend to cross-react to different analytes. But all of these can be run simultaneously in a single tube. You can get the full picture. Right? Instead of a single analyte at a time, we can get all 50 cytokines analyzed simultaneously. Right? We also have a mouse 32-plex, or a mouse 32-plex. We also have a rat 9-plex. It's been very hard to get good rat antibodies added to the panels. We also have these disease panels, and these are fairly recent, including a diabetes obesity panel with some of the more common diabetes research targets. And again, this is a full multiplexable. As I understand it, a dipinectin and a dipsin are usually run in a separate well, and the remaining are all multiplexed together. And that's because these tend to be very, very high, regardless of the health of the patient. Angiogenesis panel. Right? Again, I believe this one is run in a separate well in some cases, but not all. And then the acute phase panel, following whatever acute event, cerebrovascular event. And we very recently formatted our kits a little bit differently. The end result for you is that we've opened up a new format for our sample prep. The old beads, and I didn't even really make any allusion to it, were 5.6 micron polystyrene beads. What we've done is we've taken those same beads and we've sputter coated them, if you will, with magnetic ferrous particles. And the new bead is now 6.5 microns. It's a little larger, and it's also a magnetic bead. But it still works with the original vacuum prep, and it still runs through the same instrument. The only advantage now is we can do a magnetic-based prep, and there's some real advantages to that. Eventually, all xMAP assays will have to go to magnetic, and that's been announced by Luminex. But the reason we went for it is because there are a lot of advantages to the quality of the data. We've launched a wash station, which is both, well, it's actually magnetic, vacuum prep compatible, and it's also compatible for ELISAs. So if you want to use the same wash station to do your bead prep for vacuum, magnetic, and it will also set up and wash ELISA plates. So it automates the procedure of washing the samples. And that adds uniformity to the data and also decreases the amount of time you have to spend actually pipetting. The other big advantage, the primary reason we switched, is when you're working with the 1 micron pore size, besides being very fragile and prone to ripping, it also tends to keep things in the well that are larger than 1 micron that have nothing to do with the beads themselves. So things like hyperlipidemic wraps, you get the certain amount of material or junk still in the wells. When we aspirate that out, the 1 micron pore size keeps the junk in the well along with the beads, and that goes into the final analysis and into the instrument. Whereas if we're using a magnetic prep, we aspirate off the top and we aspirate everything that is non-magnetic. So that allows us to get all the stuff out that isn't a bead. It's also much more robust, so the little membranes don't rip because they're actually a thick sheet of plastic. The other advantage, we no longer have to worry about liquid wicking out of the wells. And that means you can store these plates long term without having to worry about evaporative loss or leakage loss. No risk of perforating membranes, no risk of well-to-well transfers, and no additional vacuum line, trap, or pump required. So we're pretty happy with the magnetic conversion. And we've only very recently switched over to the all-in-one format from 3 separate kits. And I wanted to throw this in at the end. I usually don't talk about it too much, but one of the primary applications we see people using this for is our phosphoprotein and total protein kits. This works very similar to a Western blot or to an ELISA for total and phosphoprotein analysis. So we've got a number of cell signaling signal transduction proteins as both phospho and total protein. So you can do a phospho to total ratio. Oddly enough, though, we don't have every phosphoprotein target as a total target. Where we know, we will tell you which specific residue is being phosphorylated that you're detecting. Or rather, we will tell you what residue is being detected by the antibody in the assay. We only use cell signaling technology antibodies for our kits. So we're the only company that use the cell signaling technology antibodies for the multiplex format. So if you're familiar with them, CST tends to have very high quality products. And we chose them as a business partner because they've helped us a lot with the upstream QC. So they do a lot of quality control before they send us any antibodies. As opposed to some company like Santa Cruz where they produce a lot of antibodies, but they're not always of high quality. Not all of the phosphoprotein assays are completely compatible. So there are going to be a few cases where you can't multiplex. For example, EGF receptor is not multiplexable with other targets and has to be run on its own. And S6 ribosomal protein also not multiplexable. And a few of them are not quite as multiplexable as we'd like with certain other targets. Where we have it, we'll tell you if we have human, mouse, and rat data, since these tend to be pan-mammalian. And we also occasionally have non-human primate data and dog, goat, horse, a variety of different tested animals, usually user-submitted information. When we're evaluating a new phosphoprotein assay, we test it against the Western blot. So we just want to make sure that there's a good correlation, but you can also kind of see we're picking up data that really isn't visible on a blot in a quantitative way. You can see there's a slight relationship here. It's essentially flat here. So there are some real advantages, besides the fact that you don't have a pipetter in your hand, loading these gels, analyzing them, and spending a lot of time in the imager. So they correlate very nicely with the Western blot data. So it's one system, but it's got lots of applications. Now, I only really talked about chemokines and cytokines and phosphoproteins. But a lot of these other things are out there from other companies. So if you're interested in getting the hardware or locating hardware near you for any of these kinds of applications, there's a microRNA profiling kit that does a fantastic job. It's probably one of their top products, and that's available directly from Luminex. Low-density DNA arrays, genotyping, cellular profiles. It's right now in a meet. There's a BioPlex system that is protecting you guys from West Nile. It's tasked with surveillance of West Nile virus in sentinel chickens, if you're familiar with that program, and a lot of other different things. So that's it. That's the software, the hardware, the beads and assays, and the service and support. I hope if anyone's interested in a little bit more about this technology, if they'll contact me or they'll contact Randy or Rose, my colleagues, we'll be happy to help out. Any questions?
Louisiana Tech, do you have any questions?
Yes. For the phosphoprotein, you mentioned some of them are not multiplexed. I'm not quite sure why, and based on your technology you propose.
It's a very simple reason, and that is we're having to use a pan-phosphoserine and a pan-phosphotyrosine. There are no good antibodies to detect the phosphorylated form of EGF receptor and S6 ribosomal protein. As soon as we obtain one, we'll be able to multiplex this full panel, and work is currently ongoing. We recently set up an antibody facility to make our own in Singapore. That was set up at the beginning of this year, and we're starting just now to see the first antibodies roll off the production line.
I see.
Good question.
And one of the slides you mentioned about the phosphoprotein and the total protein, and you can actually find the modification site, the residue. Do you refer to the particular amino acid as
Yes.
How was that done? Because we're actually interested in using the mass spectrometry approach, and I get curious about this approach. Yeah.
Right, so antigenically, they're distinct. We can raise antibodies against a phosphorylation, so let's see if there are any good examples here. There's not, is there? For example, P53, there's two phosphorylated residues, and they're antigenically distinct, so we raise an antibody to the portion of the protein that we're interested in testing, whether or not it's phosphorylated or not. And so there's two distinctive capture antibodies, and two distinctive, or actually the detection antibodies are in common, but the capture itself is residue specific, right, or phosphorylation site specific, right? And there are going to be a lot of cases where the interplay of the two has a lot to tell you, right, where it's not a matter of the protein is phosphorylated or not phosphorylated. There's a dominant site and a recessive site. Or at least that's what I think the literature has shown in recent years.
I see. Okay, thanks.
Any other questions for Louisiana Tech?
It's all Louisiana Tech, huh?
Right, that's, yeah.
Thank you guys for coming.
Yeah. Any questions here?
Questions. I've run the cytokine assays for probably about 5-6 years now. So as long as we've had our instrument, I've run a good bit of those. If you wanted to look into possibly setting up and designing some nucleic acid assays, haven't gotten to that point yet. Wanted to sort of on the time. It's something I still would like to do. Do you guys have protocols to sort of help with that? Okay, I'll email you that, ask for that.
Sure.
In terms of, in very simple terms, real-time PCR is basically looking at the amount of a particular gene, in one sample versus another. How does the capability of the BioPlex compare to what you might use for real-time PCR for gene expression? And I see genotyping, certainly it makes sense why that would work well. How well does it work for gene expression?
The challenge with gene expression, of course, is that it requires an upstream PCR step to amplify the material. So if you're going to do that, you could have done real-time PCR at the same time. The only thing, of course, you can't do is multiplex to that level. So there are some people that are using it for a normal versus cancer screening, where there's a particular gene expression profile for the cancerous tissue versus the normal tissue. But that's the kind of biomarker approaches are really the ones we're seeing where people are using gene expression on the BioPlex. The difficulty of design is no harder than a simple PCR setup. It's basically two chemicals, beads, oligos mixed together, and then there's an incubation, and then you run it through vacuum, and then you're done. There's even steps to do it without running it through a vacuum. We just got everything all mixed together. So it's really not complicated. And some people they prep the beads with the oligo minutes before they run the samples. And it really is that easy.
It's a pretty straightforward step as far as, okay. The other part, of course, is doing multiplexes. You still have to make sure that, like, the thing
doesn't interact.
Right, as far as creativity.
Right, and just to imagine, find sort of an empty-ish looking one. They actually sell beads. So there's your bead,
Right.
and it comes with a universal tail. So we'll call that U1. And then you, of course, just design an anti-U1, and then the rest of your oligo.
Right.
So even then, all you have to do is, this is a nice, highly specific, GC-rich sequence. And you don't even need to add the chemicals. You just sort of add the oligonucleotide.
It just fine.
And it just
So
works.
in addition to the anti-UI region, what is what length of probe basically would you typically use for this?
Had some people use some really long probes, and I'm not sure I understand the application for that. But several hundred bases. And I don't know what the purpose of that is, but you can, of course, use short, 40.
Well, I think maybe for a southern blot. I mean, a long southern probe. Okay.
Yeah. I think they're looking for, I think in one case it was meiosis. They were studying crossover recombinations. They wanted to be able to show that if you stop yeast at a certain point, that the recombination event had reached a certain point.
Okay.
And studying the progression of that crossover. There's also a really fantastic technology that Bio-Rad has itself, and I think it's worth talking about very briefly. And that is, you have a bead. This is from Panomics, who were recently acquired by another company. I don't remember who. Universal tail, we'll call that U1. And you design your oligo here. And then there's another one, it's a probe sequence. Okay, and the probe, I'll call what am I calling it? We'll call it probe. A probe sequence with a tail on it, which we'll call U2. All right, and you add anti-U2 tail with, and this is the cool part about it, highly branched DNA. And every one of those branches is highly biotinylated. So you add your streptavidin phycoerythrin, and you get like a tyramide type amplification. It's a cascade event where you get 10,000 fluorescent molecules for every single binding event. And because it's specific in the same way that real-time PCR with a probe is specific, you don't need as much material. And the sensitivity on this is down to thousands of copies. You're using an LCM material. You can do 100 genes in one well, and all you need to supply are the oligos. And they supply the branched DNA. So it really is a pretty easy system. We don't sell any products for it, so I don't get a lot of time to talk about it. But it's pretty cool.
That's good information.
I think people think it's going to be complicated. Oh, no. People think it's going to be complicated, and in fact, it's really quite simple.
Any other questions? Okay, go ahead.
Have a quick question. I used the genotype, the melting curve analysis to test the genotype. I had a problem before. I had the melt peak, but the melting temperature shift a little bit.
Mm-hmm.
So I wonder, the negative control, I did that. I have no peak shows up at that particular melting temperature. So I wonder, is there any way that I can test if this is the positive results or not.
Okay, some of the other things, if you remember, we talked about 4 things that affect the melting temperature of DNA. Let's see if I can find kind of a blank screen here. There are 4 things that affect the melting temperature of DNA: the length, the GC content, or the base composition, and the ionic strength, which includes the magnesium concentration, and also the interactions between the DNA molecules, right? So your sequences are fairly uniform. What you might not have, though, if you look at your curves, you might have one that's a little bit lower than the others, right?
Yeah.
If this happens, this one has higher levels of magnesium than this one. Free magnesium, right? Because it's bound up in the DNA. The less product you make, the higher the salt concentration. And that tends to shift your melt peak. You get one that's ever so slightly shifted.
Yeah.
Right? The other thing that can happen is if you make a lot of DNA, it tends to stabilize the DNA-DNA interactions that might be happening. Right? So if you get a very small shift, the best thing to do is, do you have access to a pyrosequencer?
I'm not sure if we have or not.
One of the little Biotage
Yeah.
PSQs. That's a great way to do quickie sequencing. The instrument itself is like $80,000, so it's not a cheap first purchase. Or you can send it off for sequencing. And so you take the product and you topo TA clone it and send it off to a sequencing lab. Or sequence it yourself and check to see what the base composition is for that amplicon. But most of the time, if you see a very small shift, let's say 1 base, it will often, if it's not matching up with genotypes, will be a function of the different endpoints that you're getting for your amplification products. But you're right, a second technology confirmation is usually a good idea with these kinds of assays. There are some other things you can do, though. If you don't want to send it off for sequencing, take the product and find a restriction enzyme that cuts at one sequence but not the other. Right? You've amplified it. Make sure that the cut site has been modified. And you may have to find just the right restriction enzyme to make that work. Right? And then run the products on the gel.
I'm not sure I follow up with that. So this particular gene we're looking at is APP, and there are 2 scenarios. One is transgenic, one is wild type. So the typical melting curve we got is either just going to go up like that, or it's just flat toe in the bottom base. Because
do you have an allele-specific primer?
We do, yeah.
Okay.
But since there's no transgenic gene in the wild type, then there's no proliferation. Then it's just a flat line at the bottom. So follow up with using the restriction enzyme. How was that in play?
Well, your transgene then, in that case,
yes.
if the product is, let's say, has a single, here's your transgene. Oh my. If your product has a single ATGC here, you can find a cutter that cuts that sequence. Because it's an amplified product, if we're seeing it on a melt curve, it's an amplified product. If you see exactly 2 bands, then you know that you've got a single cut site. I mean, that's one piece of evidence. It wouldn't be knockout evidence. But if you don't, if you see 1 band at the very top, it's the same size as the original product, then you know that this sequence is not present in whatever it is you just amplified. And that's usually kind of a quick way to confirm you've got something useful. And of course, if you see 8 bands, it can't be that sequence. So, I mean, the best thing to do is to send it off for sequencing, but then you have to wait and you have time, and it tends to cost a lot, whereas a restriction enzyme digest will give you inferential data that it is what you think it is.
And for this restrictions digest, do we follow up with like a gel electrophoresis?
Yes, exactly.
Okay, right.
Yeah. And you're going to need a high percentage gel because your product's going to be very small and you're going to be cutting it in half. So it's going to be teeny tiny. You need a high percentage gel, run very slow.
Thank you.
Any other questions? Okay, thanks.
Thank you.